Welcome to our Brandeis Mathematical Biology Seminar. Today we have Kaushik from Brandeis Department of Biology. And thank you, Kaushik.
Okay, I just want to make sure that my audio is coming in loud and clear. So if in Kana, does it sound okay? Just to reconfirm.
Yes.
Yeah, okay, thanks. Okay, so yeah, I was thinking about what my talk title should be, and I was thinking about also my audience a little bit, and then I thought that what I would really appreciate feedback on is something that we've been thinking about for quite a lot of time, which is really how do systems that are so dynamic and actually interact with each other on timescales that range from milliseconds to seconds, how do they present or acquire emergent properties that are quite interesting? And those are the kinds of things that we study in my lab. And they're all in the realm of trying to understand how gene expression actually works. And hopefully this theme will become a little clearer throughout the course of my talk. So the problem that my lab is really interested in is this question of, how do cells that have identical genomes establish and maintain gene expression states that are, say, stable and heritable for many generations? So this is a situation that we know about during multicellular development, for example, where you have the embryo, which has the same genome that should then diversify and give rise to many different cell types. And what we really hope is that none of these cells actually forget who they are throughout our lifetime. And one way of thinking about disease is really, it's a situation where the cell really forgets what normalcy is actually like, and that disruption of homeostasis can be thought of as disease, and it has both genetic and epigenetic components. Now, the problem that we actually study is at the level of how DNA is packaged within the nucleus, and this involves basic proteins called histones that actually package DNA. They are repeated at the scale of about 140 base pairs with this kind of periodicity. And it turns out that you can actually mark these histones with particular kinds of modifications, one of them being histone H3 lysine 4 methylation, for example, which is associated with active transcription, and the other being histone H3 lysine 9 methylation, which is associated with gene silencing. And this is something that my lab is very interested in. And so the question becomes, how do you actually just mark DNA packaging proteins and then produce outcomes that ultimately influence gene expression? So there must be a set of proteins that can interpret these modifications by binding to them, and then recruit or repel the transcription machinery so that you can achieve these interesting gene expression outcomes. We study this in the context of Schizosaccharomyces pompi, which has a fairly small genome consisting of about 12 million base pairs or so. And it has 3 chromosomes, and heterochromatin, which is this gene silencing phenomenon that I talked about earlier, which consists of histones that are marked by H3K9 methylation, is sort of enriched at these 3 major loci, one being at the middles of chromosome, and these are regions called the pericentrometic repeats, at the ends of chromosome, which are the telomeres, and then there's a sex-determining locus in yeast, which is called the mating type locus. There's also this unusual puzzle in yeast and in other organisms as well, where rDNA, which is probably the most highly expressed region of our genomes, is also marked with H3K9 methylation, raising the possibility that H3K9 methylation might have some other types of regulatory roles in also affecting protein synthesis. So for the purposes of the talk today, there are only 2 proteins that you mostly need to know about, and one of them is the histone methyltransferase in fish and yeast, and this is a protein called CLAR4. CLAR4 methylates histones at position lysine 9 in order to establish this epigenetic modification, which ultimately produces silencing. And these modifications, as I mentioned before, need to be interpreted in some specific way, and this is done by proteins that actually bind to H3K9 methylation. And in particular, a protein that I'll be talking about quite extensively throughout the course of my talk is this protein called SWI6, and SWI6 binds with high specificity to H3K9 methylation, meaning that it can move around in the genome to many different locations, but it can identify H3K9 methylation specifically and execute this process of epigenetic silencing. So this is really a minimal parts list, and there's probably 10 or 20 proteins or so that are all involved in ultimately silencing a gene. But for the purposes of this talk, the only 2 proteins whose identities you really need to know are CLAR4 and SWI6. Okay, so this is to just give you a sense of what the more complex version of this pathway might actually look like. And these are protein names that you do not need to know, but really to appreciate that there is a complex series of protein-protein interactions that actually produces silencing. And the way we read out silencing is by using phenotypic assays, one example of which is shown here. So yeast cells normally look white on plates, but if a gene called AT6, which is a reporter gene, is silenced, cells turn red, and you can see these colonies being extremely red on this plate. If you delete CLAR4, which is the only H3K9 methyltransferase in fish and yeast cells, all of these colonies that actually appear red now turn white. You can also see that the same phenotype is mimicked in SY6 delta cells, indicating that this minimal parts list that I outlined before, consisting of SY6 and CLAR4, ultimately defines at least a minimal set of proteins that are essential for epigenetic silencing of this AT6 reporter gene that's inserted at the pericentrometic repeats. We've also sort of distilled this entire system into something that's a lot more simple, which is you can now have just the CLAR4 protein tethered at an ectopic site, and this can also lead to silencing. And even in the context of this ectopic system, where we can establish synthetic heterochromatin domains, where we can drive H3K9 methylation at a particular region of the genome, and now establish silencing that isn't in any way connected to the endogenous heterochromatin loci, such as those at the pericentromeres or the telomeres. Even in these situations, you can successfully not only establish heterochromatin, but this heterochromatin also conforms to many of the genetic requirements that have been identified at other loci. And in particular, in this particular assay, what I want to point out is if the gene is silenced, colonies turn red. And if you delete SY6, you can actually see the sensitivity of this phenotype for the presence or the lack thereof of SY6. Okay, so SY6 is this reader protein that binds to H3K9 methylation and somehow executes this process of silencing. This, of course, brings me to the way I'm sort of going to conceptualize what I will talk about, which is that a lot of what we know about these proteins, such as SY6, is actually based on in vitro measurements of protein function. And I was struck by this portrait of an unfinished horse, which really, I think, illustrates how much more complex reality actually is. And sometimes and often, I think that we're in the realm of actually simplifying biology to the extent that we don't really capture what's happening inside of the cell. So thinking of this as one of many grand philosophical challenges in biology, what I aspire to do, at least through the course of my talk, is actually tell you how measurements of proteins inside of cells, especially single molecule imaging measurements, can actually illuminate aspects of protein function that are quite different from what we actually learn about when we study these proteins in test tubes. And this is to give you some sense of what that vision plays out to be like, which is to say that there are a bunch of proteins that actually interact with H3K9 methylation, and there are many of them. Again, the names of these proteins are not important, but what is essential to really know is that many of these proteins that bind to H3K9 methylation and interact with this locus do so on the extreme left of timescales in biology, so to speak, because they actually interact with chromatin on this millisecond to second timescale, but the phenotypes that they produce, which is affecting gene expression. So for example, these are lineages of yeast cells, which are differently colored for a variety of reasons. But it suffices to say that these phenotypes are actually an outcome of how these proteins actually bind to this H3K9 methylation modification. So we can immobilize individual fish and yeast cells within microfluidic channels. You can watch what happens within individual lineages, and somehow these millisecond to second scale dynamics that happens on the left end of this time scale really produce these dramatic silencing outcomes that are maintained on timescales ranging from hours to days. And what I hope to do in my talk is to give you some insight into how we can understand how this process works. Okay, so my talk will have two parts, and the first part I'll actually tell you how SY6 actually finds H3K9 methylation in the nucleus, and in the second part of my talk I'll actually tell you what SY6 does when it does find these sites of H3K9 methylation. The first part of my talk is work that we published a few years ago, and in the second part I'll give you some insight into unpublished data which relates to the mechanical roles of SY6 in regulating nuclear function. So most of this work was done by a former postdoc in my lab, Saikat Biswas, who's currently at PacBio, a former graduate student in Julie Bettine's lab, who's my collaborator at the University of Michigan, and Z1, who's the graduate student who worked with Saikat, is currently a postdoc at UC Berkeley. Okay, so as I told you before, SY6 is the is going to be the star of this entire talk, and it's useful to know what the hero of the story actually looks like. And SWI6 basically is quite a simple protein. It's a small protein, about 38 kilodaltons in molecular weight. It forms a dimer. The dimerization is regulated by this domain called the chromoshadow domain. There's a highly flexible hinge region, which is enriched with lysines and arginines, binds non-specifically to nucleic acids based on in vitro data. And then there's the business end of the protein, which is a chromodomain that binds with high specificity to H3K9 methylation. Based on in vivo studies or imaging studies of SWI6, SWI6 predominantly localizes to the pericentromeres, which adopt this. So chromosomes in fish and yeast adopt what is called a Rabel-like configuration. And for historical reasons, that's the name this configuration is given. But the pericentromeres are found at the apex of this nucleus. And the telomeres actually cluster on the left and the right sides, giving you 3 loci at which SWI6 localizes. So typically in fish and yeast cells, we'd see about 3 to 4 foci of SWI6, which can localize to the pericentromeres and the telomeres. The pericentromeres also have this interesting property where the spindle pole body, which is the equivalent of the centrosome in mammalian cells, actually interacts with microtubules, as a result of which the centromere is constantly experiencing forces that microtubules actually transmit to the nucleus. And in the second part of the talk, you'll actually see what the effects or outcomes of these forces being transmitted to the nucleus of fish and yeast actually is. Okay, and HP1 proteins, as I said, form these clusters, but these clusters also have interesting biophysical properties. So in fish and yeast, SWI6, as I said, localizes to the centromeres and telomeres and forms these foci, which all have liquid or gel-like properties. And at least at a superficial level, which is to say at a qualitative level, these proteins, SWI6, HP1A as it's known in flies, and HP1 alpha as it's called in mammals, have very similar biophysical properties, which is to say that they can self-associate, undergo oligomerization, and then they can actually form condensates and complex with chromatin that have liquid or gel-like properties. And it's thought that this particular feature of SWI6 or HP1 proteins can affect gene silencing. Okay, so in vitro, what we know about SWI6, and this beckons back to this the portrait of the unfinished horse that I was actually talking about, which is to say that a lot of what we actually know about HV1 proteins and SWI6 primarily come from in vitro studies of this protein. And what we know is that HP1 proteins, of which SWI6 is an archetypal member, form dimers, and about 80% of these proteins come out as dimers if you do size exclusion affinity purifications. They bind to nucleosomes with micromolar affinity or low micromolar affinity, and at least in vitro, they exhibit fairly low specificity, which means if you provide SWI6 with a methylated versus an unmethylated nucleosome, it only exhibits about a 3-fold preference for modified nucleosomes over unmodified nucleosomes. And this is sort of what we know about what the nucleus actually looks like. It's a very complex and crowded landscape consisting of many different proteins. So it's really tightly packed with a lot of different proteins and a lot of different RNA. So what we're actually trying to learn about SWI6, I would argue, is under very dilute solution conditions where the behavior of the protein can actually be quite different from what the protein behavior might actually be like inside of the nucleus. So what we use, and when I say we, I mean Saikat, who worked with Julie's lab in order to actually do single molecule imaging. What he did was to actually build these strains where PAM cherry is fused to the N-terminus of SWI6. This is a special protein which you can activate with 406 nanometer light. These proteins are dark until you provide an activation pulse. And once you provide this activation pulse, you activate typically about 1 to 2 proteins. And then you can image this protein until it undergoes photobleaching using a 561 nanometer light source. So here you can actually see an individual molecule that's been photoactivated using 406 nanometer light. It moves around in the nucleus in a heterogeneous manner until it photobleaches after a few seconds. So it's useful to think about what the conceptual problem of actually analyzing such data might actually be, which is to say that proteins may take short or long steps. And so this is completely simulated data. This has nothing to do with the protein that's under investigation here. But you can actually see that sometimes proteins make these large jumps, which we call as track length. And the track lengths can be either long or short. But this has nothing to do with what the diffusion state that's actually associated with the protein might actually be, meaning that is to simply say that long steps may actually still correspond to molecules that adopt a low mobility state, and short jumps may still correspond to molecules that adopt a high diffusion state. And this is useful to think about because it means that having an unbiased way to classify steps and also interrogate what their mobility states might actually be would be a very useful analytical tool. So this is essentially what Julie's lab had done at this point. So they developed this algorithm called SMOG. SMOG is based on a Bayesian algorithm. And what they do is actually they analyze the data of these proteins moving around inside the nucleus and extract 4 important features. The first is, what are the number of mobility states that are required to define the movement of this protein or all of our data after a single molecule tracking experiment. The second is, what's the average diffusion coefficient that's associated with each one of these mobility states? The third is, what's the fraction of molecules that's present in each state? And this is quite important because if there are multiple states and we make genetic manipulations to these yeast cells, we can actually see shifts in different mobility states, which gives us insight into what the biochemical properties that are associated with each biophysical state might be. And finally, we also get information about what's the probability of transitioning between any 2 states might actually be. And I'll give you some insight into how each of these features of the data that emerges from SMOG analysis was quite useful in understanding how proteins actually find their targets inside of cells. Okay, so if we actually take a lot of data and watch a lot of individual SWI6 molecules moving inside the nucleus of fission yeast cells and analyze this data using SMOG, what we get as an output are 4 mobility states as shown here in this plot. You can see mobility states in purple, green, red, and blue. On the x-axis, on the y-axis is the diffusion coefficient. So this represents the fastest diffusion rate that we can measure, and this represents molecules that adopt the slowest diffusion state coefficients that we can measure. On the x-axis corresponds to the fraction of molecules that are in each of these mobility states. So for example, here you can actually see that the molecules that adopt the slow mobility state correspond to 20% of our entire population. And then there's something in these green and red states, which we classify as intermediate diffusion states. And of course, then there's this fast diffusion state. The fast diffusion state is limited by the time resolution of our camera. And actually, we don't measure the fast diffusion state very well. But our cameras and our microscopy approaches are well adjusted to actually measure everything in the intermediate and the slow regime. This is not so important for thinking about this data, but it's useful to know how well we're actually measuring these diffusion states. And the most ambiguity is actually in the fast state, not in the slow states. Okay, so how do we actually use yeast genetics to identify what the biochemical properties of each of these mobility states might be? We actually do something that's very simple in yeast genetics, which is delete proteins that are important for particular biological functions. So in this case, we can delete CLAR4, which erases all the H3K9 methylation that's available in the fission yeast nucleus. And this immediately results in data not with 4 mobility states, which is the crossed hatches that are shown, which corresponds to the wild type situation. But in CLAR4 delta cells, what you can actually see is about 10% of molecules that adopt an intermediate diffusion state, but a complete loss of molecules in this blue slow diffusing state. And then, of course, a majority of molecules that dissociate from this state now adopt this fast diffusion coefficient. So this is only to say that we can make a series of deletions of this kind and actually map what the biochemical properties of each of these diffusion coefficients might actually be. Something that's trivial that we can do based on just the CLAR4 deletion is to just say that the lowest diffusion state, the slow one, corresponds to SWI6 that binds to H3K9 methylation. There's a non-H3K9 methylation dependent component that's embedded in this red state, but the green and the purple terms are actually completely H3K9 methylation independent, and in fact increase in proportion when you delete CLAR4. So this is to summarize a lot of different data, and I'll tell you one vignette based on what we learned from actually acquiring such data. So what we learned from making a bunch of mutations in yeast is that these 4 mobility states actually have distinct biochemical properties that drive their occupancy. So the fast diffusion state corresponds to proteins that diffuse rapidly inside the fission yeast nucleus. They don't bind or interact with many things. The second state, it turned out, was actually predominantly driven by nucleic acid binding. The third state, which is colored in red, corresponds to chromatin sampling, which is to say that a protein can choose to bind to both chromatin that is H3K9 methylated and that is non-H3K9 methylated. And what we're really measuring in the red state is the ability of these proteins to bind specifically and selectively to H3K9 methylation. And in the blue state, of course, what I showed you quite trivially in the situation by deleting CLAR4 is that if you eliminate all H3K9 methylation within the nucleus, then you get a complete loss of the blue state, as a result of which this blue state must be dependent on H3K9 methylation for its binding. So we can also extract transition states, transition probabilities between different states. So here we can actually see that molecules in one state can transition to another state with some definitive probability. And all I want you to actually take away from this plot is that SPY6 molecules are most likely to transition between adjacent mobility states rather than non-adjacent mobility states. And that's best illustrated by the fact that you do not see a lot of cross arrows on the diagonal, but you actually see mostly nearest neighbor transitions. So what this also means for us as thinking about the problem as geneticists is how can we actually manipulate the occupancy of proteins in each of these states such that we can either drive more proteins to the slow state or more proteins to the fast state. And I'll show you one synthetic tool that we developed in order to actually answer this question. So what we do know about SPY6 is that it forms monomers, oligomers, and condensates based on their in vivo properties and the fact that they are quite dynamic. Okay, so now what we don't know though is how many SPY6 molecules does it actually take to discover sites of H3K9 methylation inside cells. The fact that we knew what the different diffusion coefficients actually map to means that SPY6 is constantly competing with nucleic acid binding, chromatin sampling states before it actually binds to H3K9 methylation. So our question was quite simple, which is what's the minimal functional unit of SPY6 that is required to bind stably to H3K9 methylation as it undergoes these biophysical transitions from oligomers to condensates. So one thing that we learned is that the business end of the protein is, of course, the chromodomain. And if you mutate the chromodomain, it inhibits H3K9 methylation binding. As you can see, we get a result that is very similar to deleting CLAR4 in these cells, a complete loss of molecules in the blue state, and an increase in the molecules that are in the unbound or mobile states. We can do similar mutations in the dimerization interface of this protein. So you can actually mutate the fact that these proteins form dimers, as a result of which you can make a CSD mutant. This results in the protein being predominantly a monomer. And here you can actually see, again, a complete loss of H3K9 methylation binding, and again in the highly diffuse state of molecules. So what this really means is that we need to somehow figure out how many chromodomains does it actually take for these proteins to bind to find sites of H3K9 methylation inside the nucleus, and what is the oligomerization state of this protein that is required also to be able to do this. So to answer this question, we started creating artificial versions of SPY6. And one way to do this is to actually take the oligomerization domain of this protein, chop this up, and actually use a glutathione S transferase tag. This particular tag forms obligate dimers, as a result of which you can see in this cartoon representation here, if you express GST fused to a single chromodomain, it's always going to actually form a dimer. But what's interesting about GST is that it's such a soluble protein that the GST homodimer does not actually form higher order oligomers, and we were able to do additional biochemistry experiments as well to confirm this. Okay, so what happens if we actually express 1xCD GST, which is proteins that only have one chromodomain that can dimerize only to form a dimer of SPY6 molecules? And what you can actually see here is data from wild-type SPY6. You can actually see what I told you earlier about these proteins. They localize to the centromeres and telomeres, giving rise to cells that display 3 to 4 foci in each cell. However, if you express this 1xCD GST, despite its ability to recognize and bind H3K9 methylation, given that this chromodomain is intact, is unable to now bind to sites of H3K9 methylation. So the outcome of this experiment is fairly simple. What it tells us is that a dimer of chromodomains, having 2 chromodomains only, is insufficient for proteins to actually identify and bind to sites of H3K9 methylation in the fissionase genome. So Saikat then did something simple after this, which is to actually just add another chromodomain. And this was largely driven by our understanding of how this protein works, which is to say that SY6 undergoes homo-oligomerization in order to form larger structures. And we envision that perhaps having two chromodomains alone is insufficient, and the ability of this protein to homo-oligomerize might actually be important for its binding function inside the nucleus of cells. And this is one way in which cells could ensure that SY6 binds with high specificity to sites of H3K9 methylation in the genome. So here you can actually see this is the same construct, except it has one extra chromodomain. And this construct now ends up actually being able to bind to the same sites of H3K9 methylation as SY6 does in Fissionese cells. So here you see about 2 to 3 foci per cell, and you can see that foci formation is fully restored in the case of this 2xCD GST construct. However, something that was really interesting that came out of the single molecule data is that while SY6, which is shown in this, while the diffusion properties of SY6 shown in these crossed hatches shows about 20% of molecules in the bound state and about 30% of molecules in the chromatin bound state without H3K9 methylation, the 2xCD GST construct actually has a large number of proteins that are actually chromatin bound rather than chromatin unbound. So what you can actually see is that having proteins that are loosely oligomeric allows proteins to actually exist both in the unbound and the bound state, as opposed to being predominantly bound to chromatin, as is the case with 2xCD GST, which really has the inability to, which we are unable to regulate in very specific ways. So the model that we came up with was really something that was driven by our artificial constructs, which is to say that multivalent recognition is important for SY6 to bind to H3K9 methylation in cells. This multivalent recognition is not infinite, which is to say that these oligomers, in fact, only need a finite number of chromodomains to recognize and bind H3K9 methylation. And we found that there are 4 chromodomains that are required for H3K9 methylation recognition in cells. It turns out that nucleic acid binding in data that they did not show you completely antagonizes this process of oligomerization. And it's useful to remember that in the nucleus, there's a large excess of nucleic acids, and as a result of which these oligomers are constantly falling apart and turning into dimers inside the cell. As a result, these are things that are very hard to actually find out in the context of in vitro measurements, because you can't achieve the same types of nucleic acid concentrations that these proteins encounter inside of living cells. Okay, so this brings me to the end of the first part of my talk, but what I'll tell you in the second part is actually how we've used this information about SY6 and its ability to form condensates, but I'll pivot to actually thinking about its emergent properties in regulating the mechanical stiffness of Fissionese cells. So the second part of my talk is really focused on what does SY6 actually do once it binds to sites of H3K9 methylation. This is work in collaboration with Megan King's lab at Yale, and all of the nuclear stiffness work was done by Jessica Williams, a former graduate student in Megan King's lab, and Ivan Suratsev, who's a research scientist still in Megan King's lab. And the parts that were done in my lab primarily concern the single molecule tracking in collaboration, of course, with Julie Bettine's lab, as I told you before. So just again to remind you, SY6, much like its homologs in other organisms, such as flies and mouse, undergo homo-oligomerization in order to form these condensates that have liquid or gel-like properties. If you actually look at this low-resolution picture, unfortunately, in this image, let me see if I can actually tell you what you need to look at. Okay, so hopefully you can actually see these clusters. These clusters actually correspond to SY6 GFP bound to pericentromeric repeats. And so these clusters appear really bright. What's shown in these different frames is actually time. And so what we're looking at is different frames of movies across a 100-second video that Ivan and Jessica acquired. So what you can actually see, hopefully, is that these SY6 clusters within the pericentromeres undergo splitting and can fuse again. And this is primarily driven by the action of microtubules on the spindle pole body. So again, to remind you, the spindle pole body is equivalent to the centrosome in mammalian cells. It's the microtubule organizing center in cells. this spindle pole body is in close association with the pericentrometic repeats, which is the place where SY6 binds. The spindle pole body is constantly exerting forces on the nucleus of fissionese cells, as a result of which these clusters are constantly splitting and refusing. And so one sort of hypothesis that emerged from this observation was, could force be transduced from the spindle pole body to heterochromatin, and can that regulate how stiff the nucleus might actually be? So something that Megan's lab did was to do this experiment where they added 1,6-hexane diol to cells that express GFP SY6. So here you can actually see clusters of GFP SY6. As I've shown you before, they form these really beautiful puncta. There's about 3 to 4 foci per cell. If you add hexane diol, what you can actually see is that the brightness of these foci substantially reduce, but there's a bunch of SY6 that's still bound to H3K9 methylation. So in conjunction with the single molecule data that I showed you before, this very likely corresponds to the bound fraction of SY6, and what 1,6-hexane diol rapidly depletes is that fraction of SY6 which is unbound from chromatin. So Megan's lab also went on to characterize what the properties of these clusters upon addition of 1,6-hexane diol might actually be. And there's a very striking phenotype that emerges in these cells. So TAS1 is a protein that binds to telomeres. And so it's easy to actually know whether the telomeres are clustered together or not by simply adding a fusion of GFP to TAS1. And then you can actually count how many telomeres exist, or how many foci that correspond to telomeres exist in each cell. And it turns out that if you add 1,6-hexane diol, the telomeres actually decluster substantially. And the same declustering of telomeres also happens if you delete SY6, meaning that SY6 is really organizing telomeres to form specific clusters. It turns out that you can actually have mutants of SY6. This is a mutation in the N-terminal domain of SY6 that's involved in nucleosome binding. And it turns out that if you make a mutation of SY6, you can also see the same telomere declustering phenotype, which is to say that if you delete SY6, you see a telomere declustering phenotype, where telomeres which are clustered together typically start unclustering from each other. There's about 6 telomeres per cell, so we should definitely see something that's more than 2 foci, which is typically what one sees in wild-type cells. The telomere declustering phenotypes are quite acute in the case of the SY6 SM1 mutant, which got us thinking, what are the biophysical properties of SY6 SM1 that are leading to these telomere declustering mutants? So we performed single molecule imaging experiments, and this is the mutation again of this SM1 allele. There are 2 mutations, in fact. One is an aspartate to glycine mutation, and a deletion of a glutamate residue, delta E74. And these 2 mutations actually lead to a substantial reorganization of the mobility states that are associated with SY6. So what happens in the SY6 SM1 mutant is actually a severe depletion of the unbound and mobile population, but a significant increase in the H3K9 Me2 and Me3 bound population, and also unmethylated H3K9 associated proteins. So this is to actually say that this is quite reminiscent of the hexane diol observations, which is somehow SY6 SM1 has a large proportion of proteins that's stably bound to chromatin rather than having proteins in the more mobile or fluid phase. Okay, so what it turns out is that we attributed the defect in SY6 SM1 to the telomere declustering phenotypes, to its inability to actually form condensates inside cells. And at the very least, our single molecule imaging data supported the idea that there's a large fraction of proteins that are bound rather than unbound, which is very likely to affect the fluidity of these condensates. We can also calculate the transition probabilities across these different states. As I told you, each of these states corresponds to different diffusion coefficients, and SY6 predominantly transitions between adjacent mobility states. But what happens in the case of the SY6 SM1 mutants is significant non-adjacent transition probabilities that I actually see, and a large population of molecules that now reside in both the chromatin bound and the H3K9 methylation bound states. Okay, so it turns out that we can also measure the stiffness of isolated Pombe nuclei, which is important to actually think about the connection between what happens if you actually perturb the properties of SY6, and then actually look at how perturbing these properties actually affects the stiffness of the nucleus in cells. These measurements can be done by immobilizing the nucleus on a glass cover slip, linking the nucleus to two beads, one of which can be trapped using an optical tweezer. And then you can actually displace the piezo stage, as a result of which you actually pull the bead away from the center of the optical trap. And then you can measure the displacement of the bead from the optical trap. But then, of course, the nucleus is now going to exert a force in the opposite direction, as a result of which you can actually calculate what the stiffness of the nucleus is and what its viscosity might actually be as well. So these measurements are summarized here. So what you can actually see is measurements of nuclear stiffness done across various oscillation frequencies of the optical tweezer. So the way this measurement is done is to pull the bead pull the bead that's actually immobilized on the nucleus by a range of about 60 to 100 nanometers, but do this at various frequencies. And so what's shown here is the oscillation frequency response for wild type cells. For CLAR4 delta cells, you can see that it significantly decreases, SY6 delta decreases, and SY6 SM1 decreases. A summary of this data is a measurement of the average stiffness in each of these genetic backgrounds. What you can actually see is that the nuclei get softer if you deplete them of H3K9 methylation or SY6 or introduce the SY6 SM1 allele. So in fact, completely eliminating SY6 seems to make the nuclei softer. Having this SM1 mutant, which has a larger fraction of molecules in this chromatin bound state, also has the same molecular phenotype. So as a result, what we learned here is that proteins that are actually involved in heterochromatin assembly and their biophysical properties substantially affect other nuclear properties such as stiffness. So so the last question that I actually want to talk about before I end my talk is, how does nuclear stiffness in fact affect gene expression? So this low resolution slide is not so important for your understanding, other than to actually say that what we want to actually investigate is, how does gene expression get affected if we induce or introduce proteins or alleles of SY6 that have different condensate-based properties? In order to measure its impact on gene expression, we use this system that I introduced earlier in my talk, which is we can measure whether genes are silenced or not by tethering CLAR4 directly upstream of a reporter gene called AT6. Colonies turned red. If you actually add tetracycline, this protein dissociates, and then we can measure whether the epigenetic state that was induced by tethering CLAR4 can be maintained for many, many generations. So you have one genetic background where we actually see loss of maintenance, and here you have a different genetic background where you actually see a gain of maintenance. It turned out that you can also investigate what the in vitro properties of SY6 are and how it actually forms droplets outside of cells. And this is work done by Geeta Narlikar's lab at UCSF. And here you can actually see that SY6 forms condensates outside of cells. If you disrupt the dimer interface, it doesn't form condensates. But if you make oligomerization mutants, you actually form condensates with increased wetting properties. So Melissa, a graduate student in my lab, introduced these mutations into this memory strain in order to investigate whether these oligomerization mutants actually have defects in either the establishment of heterochromatin or its ability to maintain memory across many generations. So here you can actually see that the cells look equivalently red on either in SY6 plus cases or in the case of the SY6 oligomerization mutant. But if you add tetracycline and actually investigate this question of memory, the SY6 loop X mutant, which is defective in forming condensates, actually completely loses its ability to maintain epigenetic memory. And this is also reflected in H3K9 methylation measurements using chromatin immunoprecipitation that Melissa was able to do. So this is in effect to say that mutants of SY6 that affect its ability to form condensates not only affect properties such as nuclear stiffness, but they also have a profound influence on the ability of these cells to maintain epigenetic memory. So to summarize, what I told you in the first part of my talk was single molecule imaging measurements about how proteins move around in cells and how we've been able to use single molecule imaging as a tool to learn about the biophysical properties of these proteins and their movements. I also told you how the biophysical properties of SY6 condensates actually influences nuclear stiffness, going well beyond their properties of not just regulating gene expression. As I showed you in one case, the ability of these proteins to form condensates is not just important for maintaining nuclear stiffness, but also affecting epigenetic memory. With that, I'll end my talk with acknowledgements, as I've acknowledged most of the people who've done the work, which is Saikat, Ziwan, Josh, graduate students in Julie Bettine's lab. Ali was someone who did the modeling in Lydia Fredolino's lab. And the data that I showed on the memory in gene expression was obtained by Melissa. I also highlighted collaborations with Megan King's lab at Yale, and a lot of this work was actually funded by the NSF through a grant called the Understanding Rules of Life. And with that, I guess I should end my talk by, of course, today being Feb 14th and also giving a talk in the math department, I thought it was useful to actually say something about the number pi. And I will leave you all with that last parting thought and happy to take any questions.
Thank you, Kaushik. Do you have any questions?
Thanks, Kaushik. That was really interesting. I had maybe one thing related to this second part of your talk. So do you know, in terms of the mechanism of this increased stiffness? I mean, you had mentioned that these aggregates are kind of liquid-like or gel-like, and I guess depending on which of those it is, you might have a different hypothesis for what is causing the stiffness increase.
Yeah, so what I can say is that, so we haven't done in vitro measurements of the properties of these condensates. So there's something that I can highlight that, so we thought that it was more useful to actually think about the biophysical properties of these condensates based on in vivo measurements rather than in vitro measurements, because the surrounding cellular components can affect whether phase separation occurs at one regime or another. So what I can say is that the SM1 mutant, which exhibits decreased nuclear stiffness, has a larger proportion of molecules in the chromatin bound state rather than the unbound state. So if I had to speculate, what I would say is that the SM1 mutant has decreased fluidity, so it's more likely to behave gel-like rather than liquid-like. But that would be, I guess the question really beckons whether you're asking based on, what would the protein behave like in vitro versus in vivo? So my conservative answer really is based on this data. I think SM1 just has this phenotype where it has more bound rather than unbound populations. And of course, the force transmission sort of, I think, mechanism is completely mysterious.
And then I guess one other question, more, I guess this one maybe is a little bit more just naive. So in the way that these condensates are forming, I guess you see these sort of array of something that looks a bit like droplets. Do these mutations that you introduced, do these change like not just whether or not they form, but then also the number and the size of these condensates?
Yes, they definitely do. So, so in the case of, so there's something really interesting that I did not point out in great detail, is that this 2x CD GST construct, completely artificial, has nothing to do with the biology of fission yeast. Its sort of mobility state or biophysical properties are completely identical to the SM1 mutation. So what I mean to say is, again, the 2xCD GST mutant, if I were to go, well, yeah, I mean, basically it looks identical in the sense that you have this large chromatin bound population, unbound population. In that particular instance, we did count how many foci are there, and the number of foci substantially decreases. So I think that having increased fluidity leads to more foci, and somehow having a larger bound population leads to less foci of SY6 at least. Any other questions?
I have a very naive question. When the nuclei are stiffer or softer, are all the gene expression of all the genes affected equally, or is there any tendency?
I think that actually speaks to a very big question that's in the field of mechanobiology, which is how is force affecting gene expression, and are there good reporters of genes are there good reporter genes that whose response is specifically sensitive to changes in the mechanical environment of cells. And I think we don't have such good reporters. So I can answer it the other way around, which is to say that this SM1 mutant, which has this failure to maintain mechanical stability and makes nuclei softer, leads to a loss of epigenetic memory. So this is one situation in which we found a mutation which affects epigenetic memory, and it turns out it also makes nuclei softer. So the nuclear stiffness assays are also very hard to do in a high throughput manner. So what I'm thinking is, I guess the long-winded answer to your question is, we don't have such genes. But on the other hand, if we find mutations of proteins that affect memory, maybe they will all have these nuclear stiffness phenotypes. So we can sort of get at the question the other way around, which is if we find genes that are interesting, we can go back and ask the question, do they affect nuclear stiffness?
So another thing I couldn't really get it was, is the nuclear stiffness, is there a causal relationship between nuclear stiffness and epigenetic memory?
So I mean, I'm going to say yes, only based on our data, but there is no, so there is a mutation. So this is kind of what I was getting at, is there's a mutation that we identified that has a nuclear stiffness phenotype. And the same mutation also has an epigenetic memory phenotype. And hence, I'm inclined to think that that this is one example of a situation where nuclear stiffness does affect gene expression. But to your broader question of are there many genes whose expression is affected by nuclear stiffness, that I do not know the answer to.
I see.
I don't know. I feel like Thomas thinks about these kinds of questions, so he probably has a better answer than I do.
I'm afraid not, no.
I've never thought about nuclear stiffness, so it's really interesting and exciting. Do you have any other questions? Right, if not, let's thank Kaushik again.
Yeah. Thank you again for the invitation. It was fun to talk about this.
Thank you
Thank
for
you.
the exciting talk.
All right. Bye.
Bye, thank you.